Hello, everyone. Today we're going to talk about a very special type of language, a type of talk between bacteria. And that bacterial talk has a fancy name, which is called quorum sensing. What is quorum sensing? Quorum sensing is a type of language through which bacteria can communicate with each other. They actually do not talk like human beings do, but they talk through chemical molecules. Those chemical molecules can be identified by the bacteria. They are produced by the bacteria. They are identified by the bacteria. And these chemical molecules, in addition, can carry out specific tasks in the cell by binding with specific receptors. So, how do bacteria in particular communicate with each other? Because we know from our study of the cell that there are several types of communication between the cells, whether they are cell-to-cell communication, they are physical communications, or they are chemical communications. So in this video, we are going to talk about the chemical communication between the bacteria, which means that the language will be chemical molecules. This type of chemical communication between the bacteria is important to carry out several functions. The first one, it is important for regulation of the population size of the bacteria, meaning that this type of communication between the bacteria allows them to detect when they are too many. And when they are too many and their density, the number of bacteria per unit volume is huge, then the bacteria start stopping division. And this is how they regulate population size through those chemical molecules. Of course, we will explain in detail how this occurs. But quorum sensing doesn't only allow the bacteria to regulate their size, but it also allows them to regulate their behavior, meaning that if they are going to be virulent, to produce toxins, to harm a human host, then they will do the opposite. They will not start secreting that toxin when they are very few, because this would be inefficient with a huge host, but they will count themselves through these chemical molecules and wait until they are numerous enough so that if every bacterium produces a small amount of toxin, then this huge number of bacteria will have at least the minimum number of toxin molecules that is enough to harm the host. All this language and this type of communication called quorum sensing depends on specific molecules that are called auto-inducers. These molecules are considered signaling molecules. Why? Because we will find that they will bind to specific receptors in the cell and initiate a cascade or a series of chemical reactions, just like any signaling molecule we have studied before in the cell. Why are they called autoinducers? Because they are produced by a cell and capable of stimulating the same cell. So inducing means triggering or stimulating. An autoinducer means triggering the same cell that produced it. So I produce an inducer, and that inducer binds with certain receptors inside me to trigger me to do a certain function. So an autoinducer, as we said before, is a signaling molecule because it's capable of binding with receptors and triggering a cascade or a series of chemical reactions that end up by producing a response in the cell. Their main functions, as we said before, here we are recapitulating what we said. They allow the bacteria to detect any change in their population density, and it also allows them to respond to such changes of density. What kind of response do we have? We have mentioned already two broad types of response. If the density is too high, then the bacteria will stop or will slow down their mitotic division. This is one example. And the other example is that if they are too many and they want to produce a certain substance, like a toxin, for example, or a bioluminescent pigment, a chemical molecule that gives light at night, or they want to reproduce to form a biofilm, then they will wait until they are numerous enough to carry out this function. The main idea is that those types of bacteria that are quorum sensing, and many bacteria can do this, produce the autoinducer, release it outside the cell, and these autoinducers are a function of the number of bacteria. When we have too many bacteria, we have too many inducers. When we have fewer bacteria, we have fewer inducers. So the autoinducer number of molecules is directly proportionate to the number of bacteria that produce it. All this will be clarified in examples. Why is this done? Why do bacteria want to estimate or evaluate their population density? We are repeating ourselves. First of all, they want to regulate their population size, as we said. When there are too many, they want to know, they need to know that they are too many, and therefore take an action, which is to slow down mitosis. And on the other hand, we have the opposite. When they want to produce a certain substance, it's only beneficial when they are many. If they want to produce a pigment, if they want to produce a toxin to be virulent and oppose the immunity of the body, then they have to wait until they are numerous enough to secrete a considerable amount of the pigment or the toxin at the same time. Because if only a few bacteria every now and then produce the toxin or the pigment, then it would be inefficient. It has to start at the same time and end at the same time, so that the bioluminescent pigment or the toxin are concentrated enough and their amount is high enough to achieve the function. We have discussed two examples. The first one is bioluminescence. Bio means biological, and luminescence means fluorescence in night. So there are certain bacteria that live with the squid in the sea in symbiosis, and that produce a certain pigment called luciferin pigment. And that pigment is bioluminescent because it emits light. And the other example is the formation of biofilms, those layers of microorganisms that we don't see but that can form on any surface, like on rocks, on boats, on cutting boards in the kitchen, on doorknobs, as we've discussed in infection control. You can refer to the slides or the presentations on infection control. How do autoinducers work? They work essentially by achieving an ultimate goal. What is their ultimate goal? We have said that they bind with receptors, and when they bind with receptors, they activate a chemical pathway to achieve a certain function. But the ultimate goal of all these biochemical pathways is to regulate gene expression. And when we say regulate gene expression, we mean turning off or turning on the gene. because the gene might be present, but non-functioning, turned off, and also turned on. So what happens is that the autoinducers, as we will see in the diagram, but I'm postponing the diagram to the end of the explanation so that it could be an application to all what we have said. So the autoinducer molecules are produced inside the bacteria, and then they move out of the bacteria to the external environment, to the extracellular fluid surrounding the bacteria. In some bacteria, they move simply by simple diffusion from higher concentration inside the bacterium to lower concentration outside the bacterium, like in gram-negative bacteria. Gram-negative and gram-positive is a type of staining that helps us differentiate between two types of bacteria. So the gram stain is a dye that stains the bacteria. Gram-negative bacteria appear pink under the microscope. Gram-positive bacteria appear blue or purple. The main difference is that gram-positive bacteria have a peptidoglycan wall that is thick, and therefore when the autoinducers start to move outward, they cannot move by simple diffusion, but they move by active transport, although their concentration inside is higher. And of course, this is a bit energy consuming for the gram-positive bacteria. How, as we said before, the autoinducer concentration represents bacterial density. How? Because the more the bacteria reproduce, the more we have of autoinducers, because if every bacterium produces only one autoinducer molecule per second, then 1 million of bacteria will produce 1 million of autoinducer molecules. Now the bacteria are reproducing, and the population of bacteria are producing autoinducers inside them, and then they diffuse to the outside to the extracellular component. But there comes a time when the mass of autoinducer molecules outside reaches what we call the critical density or the critical mass. What is the critical mass? It is this mass of autoinducer outside the bacterium that becomes higher than the inside. In such a case, when the bacteria continue producing autoinducers inside, these inside molecules can no more diffuse to the outside. And if the bacterium wants them to move to the outside forcibly, it has to do active transport. And therefore, this would be very, very energy costly, in addition to the original energy cost for the gram-positive bacteria. So at that time, when we reach the critical mass outside, extracellularly, then inside, there is a major decision that is being made. We will stop producing autoinducers. When we will stop not producing, autoinducers, we will stop sending the produced autoinducers outside. But we will not, at the beginning, for the very first while, we are not going to stop producing autoinducers. We are just going to continue producing inside, but they will not be able to move to the outside. So what happens at the end of the day is a gradual accumulation of the autoinducers inside the bacteria because they have been simply prevented from diffusing outside. At that moment, when the autoinducers find themselves doing nothing, incapable of going outward, they start finding a job inside the bacterium, and they start binding to specific receptors, specific to it, that are complementary to them inside the bacterium. These receptors will trigger, or this binding between the autoinducers and the intracellular bacterial receptors will trigger a cascade of chemical reactions. This cascade of chemical reactions will result in an alteration in gene expression. This binding between the autoinducer and the receptor will trigger a cascade of chemical reactions that will finally result in a response. And that response is regulation of gene expression, either activation of gene expression, turning the gene on, or inhibition of gene expression and turning the gene off. The first genes to be affected are the autoinducer genes themselves, because the autoinducers are peptides and they need genes that are transcribed and then translated into an autoinducer. So these are the first genes to be affected, and those genes will be upregulated or downregulated according to the need, as we will be saying in the next slide. So the first example is how we control population density. As we said before, quorum sensing enables the bacterial cells to know or detect when their population density becomes high and allows them to respond by downregulation of mitosis, by downregulation of the genes that produce the autoinducers first, and then by downregulation of the genes that produce the protein, the famous protein growth factor that induces mitosis. So as we said before, when high densities of bacteria are present, the autoinducers will accumulate intracellularly and will bind with their specific receptors and will restrict the expression of certain genes. And by restricting the expression, we mean downregulation or turning off or slowing down the action of certain genes. So as the bacteria reproduce during their reproductive cycles, they produce autoinducers, I'm just repeating, and the autoinducers diffuse or are actively transported to the outside. Here we are recapitulating the whole thing. And as the bacteria increase in number, more and more autoinducers are produced. Extracellularly, they will become very high. They will reach the critical mass, and the cells start finding that sending them outward to the extracellular fluid is now not only useless, but also very energy costly, because we would need a lot of active transport and hydrolysis of ATP to carry out their functions. So they continue producing autoinducers, but they are not sent to the outside. The result is secondary to the increase in the extracellular concentration of autoinducers. The intracellular concentration will increase. They start binding to their own specific receptors. That's why we call it autoinduction. And these receptors, some of them are in the cytosol and some of them are in the nucleus. And this triggers a signaling cascade, a series of chemical reactions that will end by a cellular response, resulting in inhibition of transcription factors and inhibition of course, if you inhibit transcription, then you inhibit translation and you inhibit the formation of the autoinducer, and therefore you inhibit mitosis by inhibiting, of course, the growth factor, as I said. The second example is how autoinducers help modification of the behavior of the cell. We said before that certain functions are group behavior, like production of a bioluminescent pigment or dividing rapidly to form a biofilm on a wet surface. These actions will be successful only if they are done by a large number of bacteria at the same time. So the bacteria simply vote. How do they vote? They vote through the autoinducers. They count the number of autoinducers. How is the number of autoinducers counted? The number of autoinducers is counted by this critical mass. When the critical mass is reached, the bacteria find that the autoinducers do not want to go outward. And at that time, the bacteria understand that the critical mass has been reached and that it's high time now to start production of the pigment, because When autoinducers are inhibited from going outward, then we are too many. Then let's now activate more and more transducers inside, autoinducers inside, and let's now activate the genes or the expression of the genes for formation of a pigment or mitosis, a protein for mitosis. One of the major examples of how the bacteria decide to carry out a certain function when they are entirely sure that it will be successful because their number is big is virulence. Virulence means a virulent bacterium is a bacterium that is hard to resist or hard to fight by the immune system, whether because it has a very strong bacterial wall that cannot be penetrated or destroyed, or because it has certain toxins that are produced. But if the bacteria start producing toxins when they are not many, then this simply means that this would be loss of energy or waste of energy because these bacteria are very few. So they will produce very few molecules, so they will be burnt out. The body will counteract the toxin and will counteract and kill the bacteria. So they would be ineffective because the human body is huge and they know that the human body is huge. So they wait until they reach the critical mass of autoinducers. And when this critical mass is reached, they start all at the same time to produce the toxin, in which case a considerable amount of toxin is produced. So this happens in the type of bacterium called Staph aureus or Staphylococcus aureus. As you know, this is a type of spherical bacteria that have a specific grape-like arrangement and that are seen golden under the electron microscope. They have a virulence gene, a gene that when expressed, when translated, will result in the formation of a toxin. But that virulence gene is activated only when they reach a minimum threshold limit of number. They do not need to exceed that threshold. Okay, of course, the more the better, but at least we should have this minimum number that allows us to produce an adequate amount of toxins. Before this, it would be energy costly and it would be waste of energy. So each Staph aureus will secrete a small amount of auto-inducing peptide. And when the auto-inducing peptides extracellularly increase and the critical mass is reached, they will accumulate inside, they will bind to specific receptors inside, and therefore activate a gene for a particular toxin. And the toxin, in case of Staphylococcus aureus, is a hemolysin, a toxin that is capable of attaching to the cell membrane of the red blood corpuscles and resulting in the rupture or lysis. Another example, as we said, is the down regulation or negative feedback. I said before that two important functions like bioluminescence or biofilm production are produced only when we reach the critical mass. And similarly, slowing down of cell division occurs when we reach a critical mass. After having produced bioluminescence or the dividing to form a biofilm, we will not produce forever and we will not form a biofilm forever. There comes a time when it becomes energy costly and less effective, at which time the cells start what we call down regulation of autoinducer synthesis or diminution of autoinducer synthesis. And this is what we call autoinducer negative feedback, meaning that the accumulation of the autoinducer inside the cell for a certain period of time is important to stimulate the genes and help them be expressed, those of bioluminescence and those of biofilm production. But when the task is done, we will start a negative feedback, meaning that the more autoinducers produced after the task is finished will result not in forming more autoinducers, but in forming fewer autoinducers. This is called autoinducer negative feedback. The more you have of the autoinducer will result in less production of the autoinducer. This autoinducer negative feedback also occurs in another or a third condition when we reached a critical mass, when the bacteria density becomes very high and we start slowing down mitosis, again through negative feedback. On the other hand, as I've just said, during the active formation of a bioluminescent pigment, or during the active formation of a biofilm, or during the decision of carrying out mitosis to increase our number before reaching the maximum density, we are not in a state of autoinducer negative feedback. We are in a state of autoinducer positive feedback. And this is, for the moment being, important because at that moment, when we are still producing the pigment or still producing a biofilm, or we still need to divide, we need to increase the autoinducer. So the more autoinducers you have, the more they activate the genes, and this is what we call positive feedback. The positive feedback is positive feedback for producing more autoinducer, for synthesizing more of the enzyme, for production of luciferin pigment, biopigment, or biofluorescent pigment, or for producing of more growth factors that are necessary for mitosis. By having said this, we would have finished this video of, this is a new series of videos that are tackling important and difficult concepts one by one, and they're called monotopic videos or one topic videos, and they are essentially prepared to help you focus on a particular difficult concept and break it down. Thank you so much, and see you on the next video.